Okay, let's start. That's why we can finish earlier. Right. So the previous chapter was about the structure of DNA, and so what we are going to see, I think it's very beautiful because it really shows how important the structure of protein and the interactions between proteins and DNA are important for this. And the DNA replication, repair, and recombination. This is really essential for life. If this doesn't happen, life just doesn't continue. So, first we're going to see DNA replication and then DNA repair. First point, base pairing enables DNA replication. This is very easy. That's family, I don't know who they are, but although we all have DNA, my DNA and your DNA, maybe 98, 99% is the same, but there are differences, okay? There are some mutations, there are some small differences in sequence that actually results in us being the same species but being different individuals, okay? That's very important. And thanks to this base pairing, it ensures that DNA... Sorry. Oh, don't be sorry, I just started. And it shows that, DNA can be... the fidelity of DNA replication is very high, but it allows some variations. And very easily, let's start. This chapter is maybe a little bit complex, but the beginning is very easy. So in DNA replication, there is a S strand and an S prime strand, or it's sometimes called the sense strand and the antisense strand also. But in DNA replication, one strand is used as a template for the synthesis of the other strand. So for example, this S prime strand is used as a template to synthesize the new S strand, and the other template, the other strand, is used as a template to synthesize the new S prime strand. So this is really key to make sure that the two copies are virtually the same as the parent DNA double helix. And nowadays, this is something that we all know, that if you imagine an original bacteria or original cell that replicates once, twice, 3 times, then you get a dilution of the original strand. Okay, this is divided by 2, then 4, and then 8. So we all know this, but in the beginning, after Watson and Crick in the 1950s, all they had was the structure of DNA. How replication happens, they had no idea how it happened. Now we know this, but there are 3 cases. What people imagined was 3 types of DNA replication. The DNA replication that is called semi-conservative, which means in the future generation, only one strand is conserved. Okay? We know now this is correct. Okay, we all know, but how we know it's interesting. Some scientists were thinking, okay, this seems very difficult. This seems very easy. It has to be more difficult. And some people were saying, actually, what happens is that small bits are broken off and newly synthesized in both the new generation. This is dispersive, okay? So you can see that for each of the strands, it's broken off and synthesized a little bit like chaotically, like this, okay? So each future DNA strand is a mix of the old and the one. So this is dispersive. Yet another school of thought, some scientists were thinking now what we think is happening is this. actually the new generation, one daughter cell inherits from the complete old one, and the new cell is completely new, with a completely new DNA. So of course they had to make experiments to understand what's happening. And what's very easy, the first experiment actually showed that the real thing was happening was either this and this, or this. Okay. They could very easily know this does not happen. What's happening is this one or this one. So let's see how they did it. So first, you take bacteria. It's always in the beginning with bacteria because it's very easy. You get many generations very quickly. And the two scientists, this Messerson and Stahl, who actually did this experiment. So you get bacteria, but they are growing in a medium containing nitrogen 15. And another population of bacteria, they were grown in nitrogen 14. Okay. So then you isolate the DNA of this bacteria, you isolate the DNA of this bacteria, and because this is slightly heavier, so the density will be different. Okay. So you centrifuge this thing, and after that, the heavy nitrogen 15 DNA, after centrifugation, will form one single band in the centrifugation tube. And this nitrogen 14 labeled DNA is a little bit lighter, so it will stabilize in the density gradient at a lower density band here. So that's very easy, okay? But it doesn't tell us anything. The beauty of it is the next experiment. This is really nice. So they had bacteria. Hello, long time no see. So a very nice and elegant experiment, but very simple. Usually the best experiments are very simple. First, you grow bacteria in this light medium, like nitrogen 14 medium, and you centrifuge, so you get this band, low density band, right? And then, as you know from the previous experiment, if instead of growing your bacteria in nitrogen 14 containing medium, you grow them in nitrogen 15 containing medium, these bacteria will get a heavy DNA. Okay? So when you centrifuge DNA you extract from these bacteria, there will be a high density band there. But then what you do the same thing, but after 20 minutes, at the end of the experiment, you transfer these bacteria in a medium that contains light isotope of nitrogen. Only 20 minutes. Only 20 minutes. It can be a little bit more, like 30 minutes or even 1 hour, but you really want to limit the time you do that. And then, after growing for a small amount of time these bacteria in a light medium, then you centrifuge. And what you get is you have an intermediate band here. It's not the high density band of a nitrogen 15 labeled DNA. It's not the low density band of a nitrogen 14 DNA. It's an intermediate DNA. Okay. And you can understand what's happening is that the bacteria here, they only contain nitrogen 15 labeled DNA. But for 20 minutes, they could maybe only have one round of division. So their DNA is half. One copy of the DNA is nitrogen 15, and one copy of the DNA is newly synthesized, and is nitrogen 14. The problem is with this experiment, they could not know which is correct. Because either in the semi-conservative or the dispersive model, the DNA would be half nitrogen 14, half nitrogen 15. So they did not know which one was correct. They could say, okay, so we conclusively prove that the conservative model is not correct. Because if the conservative model was correct, what would have happened in this experiment? What result would you have? if the conservative model had been correct, what would you have in this case?
There are 2 bands.
Yes, you would have 2 bands in one tube. But it's not the case. You have an intermediate, one single intermediate band, which means the newly synthesized DNA molecules are composed half of 40, half of 50. Nitrogen 14 or nitrogen 50. So, either this or this is correct. What's the next step? You want to tell people that either the semi-conservative model is correct or the dispersive model is correct. And you don't have much things in your lab. You mostly have a centrifuge and you have nitrogen 15, nitrogen 14. So you can use the same thing. What would you do? What experiment would you do to show that either one of these models is correct?
Does it have something to do with mitosis? Celebrate.
Yeah. So this because there is a question, I should maybe go back a little bit. This is about going about DNA synthesis, and in DNA synthesis, what we know now is that so you have the parent DNA strands there, and always one strand is used as a template to synthesize the new strand. Okay. So, for example, in this case, the S strand is used as a template to synthesize the new S prime strand or antisense strand, and the other one is used as a template for the new S strand. Okay, and now we know that this is what's happening: is that each strand is used as a template to synthesize the new strand. So the original strands, they get progressively diluted into the population as DNA is replicated. But scientists, even after the discovery of the double helix by Watson and Crick, they had no idea that this was happening. And there were essentially 3 schools of thought. One was, okay, what's happening is this, which is the real one, we know now. But some scientists were saying, actually what's happening is that each DNA is kind of copied in a dispersive manner. So each daughter DNA is a mix of old and new one, something like this, a little bit chaotic way. And some people were saying, well, I think what's happening is more like one daughter cell inherits from a completely old DNA, one daughter cell inherits from a completely new DNA. So 3 different models. And that's the first experiment that showed that actually what's happening is not that. Okay? Because when you grow bacteria in a medium containing heavy nitrogen, and then you transfer them in a medium containing lighter nitrogen, then you get, DNA that is composed of both nitrogen 15 and nitrogen 14. So they knew what's happening. But what do you do for next? You want to show, so of course you need to use mitosis, right? You have to sort of use bacteria and see what's happening. But actually maybe you don't need another round of mitosis. Maybe you don't even need to grow the bacteria again. Maybe you can use these guys. That's a hint. Maybe you can just use these guys. Maybe you don't need to grow them again. What could you do?
If you heat DNA, hydrogen bond will collapse. So heat DNA and centrifuge, and if you get 2 bands, a semi-conservative model is correct, and 1 intermediate band was achieved. Dispersive model is correct.
That's exactly true. That's exactly what they have done. So if you heat and keep them hot so that they don't reassociate themselves. But if you heat these two guys, essentially what happens is that you will have one nitrogen 14 band and one nitrogen 15 band. But if this model is correct, then both bands will be composed of nitrogen 14 and nitrogen 15. So even though you separate the two strands, they will still migrate during centrifugation at the same location. And so when they did that, then they showed that indeed this is the real thing. Okay. And this is called the semi-conservative model, which is not a model now, it has been proven, so it's more than a model. Okay, DNA synthesis begins at replication of origin. If you remember, we saw last week that replication origin is where you pull apart the two strands of DNA and start synthesis. Okay? So this is exactly what happened. You have a strand of DNA and you have the replication origin here. You pull the two strands apart and then you get a fork like this. And the double helix, as you see, opens with the aid of initiator protein. Of course, DNA will not pull apart itself. You, again, need protein to do that. They will pull apart this. Okay? But as you know, the two strands of DNA are quite firmly attached to each other by many hydrogen bonds. You know, it's just like trying to pull apart your zip pocket. It's very hard to do that. What do you think you will have around this? You want proteins to be able to open the double strand here. You don't want to start at the end. You want to start at the center here, so that you have two forks. One fork here and one fork there. What can you imagine you will have at this location? Generally, you know, it's C, G, A, T, right? So you want protein to be able to separate the two strands. So what do you think the origin of replication will contain? What kind of sequence you will have there? What type of nucleotides you will have in the replication origin? To make it easy for proteins to pull apart the two strands.
Any type of sequence seems to be okay.
Yeah, any type of sequence will be okay. You just need to have a protein who does it, right? But usually, during evolution, cells have evolved mechanisms that make their task relatively easy. Okay. So what would be?
So like covalent bonds are stronger, so maybe no covalent bond.
There will be no covalent bond, right, between the two strands of DNA.
Okay.
There are only nitrogen bonds between guanine and cytosine and between adenine and thymine. So there is no covalent bond in these things here. But how could you make the task of this initiator protein complex easier?
Sequence of same base pair.
Not quite. Maybe more details. I need more details. You have 4 bases, right? C, G, A, T. So what bases would you expect to be located at this replication origin?
AT rich sequence.
Why?
Because A and T hydrogen bond is weaker than CG
Yeah.
bond.
So that's exactly what's happening. So usually these replication origins are AT rich because AT only make two hydrogen bonds with each other. G and C they make 3, so this is much more difficult to separate. Okay. So these replication origin will usually contain lots of AT to make it easier. And So what you do after separating the two strands, then as I said, you have the two replication forks. And then one strand is used as the template strand. So this is a template strand for this new one here. And this is a template strand for this new one here. Okay? And from the opening of the replication origin, DNA will be synthesized this way, and DNA will be synthesized in the other way. And it will go outwards, like this. Okay? But there is something a little bit strange. If I tell you there is a rule that DNA synthesis can only proceed in the 3' direction. So DNA polymerase will always elongate the 3' end. Okay? If you remember, the 5' end and 3' end is different. Okay? Is this a problem? When you see this very simple diagram or schematic, is it a problem if I tell you that DNA synthesis can only proceed in the elongation? So when I mean elongation of the 3' end, is that if you imagine one strand of DNA, so you have the 5' end usually here on the left, and the 3' end is here on the right, and then you have the opposite strand, which has opposite polarity. So you can only synthesize DNA that way. So this is the 3' end that is elongated, and you can only synthesize DNA then, therefore, that way. So you look at this one. Is it a problem? So this is 5', right? This guy here. This is 5'. Therefore, this is 3'. So this is 3', and this guy is 5'.
So when they start building new strand, they like of the upper what is it?
Come on, draw.
No.
Come. So yeah, what is this guy here? What is this guy here? Is it 5' or 3'? This guy here. Yeah, when I was at university, I had trouble with that. What is this one here? What is this one here?
C.
What extremity it is? Is it 5' or 3'?
3.
This is 3, yeah. This is 3'. What is this guy?
And 5.
This is 5, right? What is this guy?
5.
This is 5. And then what is this guy?
3.
3. All right. So, taking into account what I told you, if you look at this, what is, and I told you the replication fork, if you actually take a look at what's happening, it's that way. Okay? That way. It started in the center and then it goes like that. So, taking this into account, you've got a problem, right? What's happening?
Upper right tip and
Lower left.
left tip cannot
That's
elongate.
Right? It goes that way. So if the rule is true, the only strand that can be synthesized is this guy. and then you can make this new one here, and this new one here. So that's kind of a problem, right? Cell has a problem. So, and it's really what's happening, because if you can take a look at a fragment of DNA that is being replicated, so what's happening here, it's an electron microscopy, electron micrograph of a DNA strand that is being duplicated. And this is the graph. This one here is what you have here. So you know here you have one replication fork here, you have one here, again here, and here. What are these little dots that you see here all along the DNA? They are not represented here. Okay, these only represent the way it goes. So this replication fork will go to the left, this replication fork will go to the right, and so on and so forth. But we can see stuff here, right? These little black dots. Right? What are these black dots?
They wrap around.
Yes.
Looks like candies.
Temaki. That's what you are eating. So what is this temaki? What does the history temaki? So what are these things? Do you remember the name? Nantoka so, ne? Whatever so it is. Nucleosome. These are the nucleosome, and so this is the DNA that is wrapped around the histone core. Okay, so this is what you have. So this is really what's happening, right? The fork proceeds, so this is the origin of replication here, and so it go in this movement. But then what happened with the 3-5' rule? You know, this is rubbish or this is true? DNA polymerase synthesized DNA using a parental strand as template. No, shit, Sherlock. So this is what you have here. As I told you, you can only synthesize DNA in the 3' end. Okay, so you add nucleotides that way, using this strand as a template. All right, and that's actually why you do it, because here you have the OH end of the 3', and you have a new nucleotide triphosphate that comes, and you use the energy that you get by cutting this pyrophosphate out. Then you get a lot of energy that allows you to make the bond between the new nucleotide and the elongating 3' end. And this is what's happening. And this is the polymerase, DNA polymerase, that is just, you know, straddling the DNA. And you have a more cartoonish way to show that you have nucleotide triphosphate comes here, attached to the 3' end, and make a nice new bond between its partner. Okay, so that's what you get. The replication fork is asymmetrical. So this is again what I told you, direction of replication fork movement, but we have a problem. Actually, we have no problem. Because what's happening to allow this to happen, sorry, my English is just going bad. So this replication fork will go that way, and this replication fork will go that way. So of course, only this strand will be able to continuously be synthesized in the 3' direction. This strand will also be continuously synthesized in the 3' direction. But what you will have, and I will go over more details later, you are having what is called Okazaki fragments. Even though the replication fork will proceed in this direction to the right, you will synthesize DNA to the left. What you get is short strands of DNA will be synthesized in the 3' direction. So, of course, you cannot synthesize that way as the replication fork progresses, but it's kind of a retrograde synthesis. Okay? So, you are the DNA polymerase, you go that way, and you synthesize backwards. So this is what's happening. Okay? And of course, you have this on the right replication fork, and you have that on the left replication fork. And you can imagine that this is more direct. It's a continuous synthesis. Okay, you elongate the 3' end. So you don't need to do any strange things. So it's quicker. So this is why this is called the leading strand template. This is the leading strand, because this is the template strand that allows a more efficient synthesis of DNA. So this is the leading strand here. So of course you have the lagging strand on the other end. And if you look at the opposite replication fork, this is the leading strand here, which allows a 5' to 3' continuous synthesis. And this is the lagging strand, because you have to synthesize retrograde, you need to sort of synthesize backward, always in the 5' to 3', 5 to 3', 5 to 3' direction, always. So we are going to see how this happens, because I think it's a very beautiful molecular mechanism. DNA polymerase is self-correcting. Yes, because if you think about it, DNA polymerase has every single round of cell division has a lot of DNA to replicate. So if it did not have these mechanisms, you would be more mutant than you are now. Okay? So the DNA polymerase actually contains two domains. There is one domain that allows the synthesis of, the addition of a new nucleotide, but there is another domain that if, for example, the wrong nucleotide is added here, what's happening is that the polymerase will actually detect, oh, I've made a mistake, and it will remove this nucleotide and try again until the right nucleotide has been added. So there are usually two domains in DNA polymerase. Okay? One that actually adds the nucleotide, and the other one that actually checks that the right nucleotide has been added. So this is again showing in bigger cartoon. So this is the polymerizing domain, if you want to actually polymerize the strand. And when there is when there is a mistake committed, then there is the editing domain that will make sure that the wrong nucleotide is removed. And this one is maybe not so interesting. I can show you if you want. It's just the molecular representation of what's happening, but it's not so interesting. So you may want to know. It's not explained in the book. They tell you that DNA replication only proceeds in the 3' direction, right? But you may want to know why. Because chemically, you could do all you need to get is energy. And if you have enough energy, you can either synthesize 3' or 5' energy. And actually, one potential reason why everything on Earth that has DNA gets its DNA replicated in the 3' direction, it's not because of DNA synthesis itself. Because actually, you could consider this is really what's happening. Okay? So you have the 5' phosphate here, and you have the 3' OH here, which is not shown. And you get energy from pyrophosphate, okay, that you remove these two phosphates from this new nucleotide triphosphate, and then you add this new nucleotide on the OH end. And so you generate a new 3' end that can be used for the next round. But you could imagine the other way around as well. Imagine if the DNA synthesis proceeded in the 5' direction, then you are using this pyrophosphate here to add the OH end of a new nucleotide, and then you generate a new 5' end with a pyrophosphate that is available for the next round. You could do that. Chemically, that's possible. However, I told you this enzyme, the DNA polymerase, has a different activity. You don't have this figure in the book. Okay? the polymerase has a proofreading activity. It's an activity that allows the polymerase to remove a previously inserted nucleotide. And because of that, then the proofreading, in the real situation, what the proofreading enzyme does is to remove the nucleotide that it just added. Right? So you remove this nucleotide with a phosphate. Up. And then you get a 3' end here, just as you had there, that can be used directly by using the energy from a new nucleotide triphosphate coming out. So that's fine, that's really what's happening. If the elongation of the chain was happening in the 5' direction, after proofreading, what happens is that, shit, I've made a mistake there, I must remove this nucleotide. So you remove it, and then you are left with a single phosphate here, and you want to put a new nucleotide triphosphate, but this you want to keep it, because you want it to be available for the next round of division. So where does the energy come from? Okay. In this case, the energy comes from this here. Okay. But in this case, the energy would come from the end, the 5' end of the chain. And then when you add a new nucleotide triphosphate, you have a new triphosphate end that you can use to add the new one and so on and so forth. But if you consider the proofreading activity, removing this thing, you will have no more energy to actually continue the synthesis. So this is one of the reasons why people think that actually you cannot have DNA synthesis in the 5' end. Of course, you could tell me, well, it's possible because you could get energy from ATP, and you could actually get ATP is used to make the bond between the two. Yes, of course, it's possible, but maybe it's more energy demanding. Okay? In this case, you don't need ATP. You just need the energy of the nucleotides you are adding to your DNA. You don't need any other nucleotides. Okay. All right. So we go back to this 3', 5' problem. Short length of RNA acts as a primer for DNA synthesis. Yes, so there is an interesting thing that DNA polymerase can elongate the 5' end. But that means that can elongate the 3' end, which means that a 3' end must be available. If you have no 3' end that is there, polymerase cannot do anything, because it needs to continue the 3' end. So, if I go back to the replication origin, if I go back there, maybe a little bit further back, this one here, you just open the 2 strands. You have no 3' end to start DNA synthesis. DNA polymerase needs a 3' end. So, it's a little bit complicated, but what the cell actually has resolved is, because the DNA polymerase cannot synthesize a new DNA strand, what you do, there is a protein, it's called a primase, that actually make an RNA primer. So this is kind of funny, because the first step of DNA synthesis is not to synthesize DNA, but it's to synthesize RNA. You are using the DNA strand as a template to synthesize a primer of RNA. And you are using a protein that is not DNA polymerase. It's a kind of a special type of DNA polymerase that has as well an RNA polymerase activity. And then you get RNA primer. Okay, so that's the first step. Remember, you always have to start DNA synthesis by making RNA primer. And then after the RNA primer, then you have an available 3' end that you can use to continue synthesis of DNA. All right?
So RNA primer contains uracil.
Yeah, exactly. It contains uracil. And there are two things. So it's RNA primer that is synthesized by a separate polymerase than the one that makes the whole DNA polymerase. Okay. This primase is not very good. It's not so good. It can make a lot of mistakes, right? But that's all right. That's all right because this is RNA primer. And the cell, because it's RNA, very easily can recognize, all right, that's the RNA primer here. I can get rid of it and make something else afterwards. So we will see the different steps that exist. But the RNA primer, the RNA primase is actually not very good enzyme. And there may be a lot of mistakes here, but that's okay because it's going to be removed later on. All you want is to provide a 3' end for a new DNA polymerase to come and synthesize the whole thing. Okay, remember this one. So what you get is this one. You imagine the first step is this one. So this is RNA primer that has been added by the primase. And then you have a 3' end that is available. After that, you have a new polymerase that comes and elongates the 3' end of this primer here. And then what you have here is the same thing that happened here. But here it already happened. So you had RNA primer and the primase went out. A new DNA polymerase came and elongated the 3' end. So at the end, the two fragments will sort of join each other. So on the lagging strand, in the beginning, you have an alternation of RNA primer, fragment, RNA primer, fragment. This is complex, right? This is what you get here in a previous slide. So here, not everything is shown. What you have here is the fragment of DNA. But you imagine in between, there are little fragments of RNA. And these fragments of DNA, they are called the Okazaki fragments. Because the person who actually teased apart the mechanism is called Okazaki, all right? So this is why they are called Okazaki fragment. But you must get rid, as I said previously, you must get rid of the RNA primer because they are not useful and they have many mistakes. So after that, you then remove the RNA primer by a nuclease. So it's a different enzyme that will remove the primers. And then you must have another polymerase, another DNA polymerase, that will fill the gap left by the RNA primer removed. And then that's what you have here. So the RNA primer has been removed, and it has been replaced by DNA with a different polymerase. And then there is yet another enzyme that must then repair this nick between the previous Okazaki fragment and the newly synthesized Okazaki fragment. So you have many enzymes all working together to make sure that DNA is smoothly synthesized. And something that's interesting is that bacteria and human, it's pretty much the same. But in bacteria, the speed of synthesis is about 1,000 nucleotide pair per second. And in human, it's about 100. Why is it slower in human? Why it is slower in human? Why the synthesis of DNA is slower in human? What could be the reason? We don't really know for sure, but can you propose a theory of why? I think I have it somewhere in the reins. I wrote it somewhere. Can you imagine a reason why?
DNA polymerase have edit area in humans.
Yeah, it could be. Yeah, you're right. It could be maybe because some polymerases were extremely, they have a very high proofreading activity. This provides a delay. But bacteria also have proofreading activity because it's as important for bacteria to keep their DNA intact than for us. So proofreading activity is actually not so different. So what could be another reason? Actually, yeah, it's eukaryotic DNA polymerase are about 10 times less fast at synthesizing DNA. What will be the reason? Think about everything you know so far. Why would the synthesis of DNA be slower in eukaryotic cells?
DNA in eukaryotic cell is very condensed, so uncondensed takes time.
Yeah, so that's one of the potential reasons, that's right. So, because the DNA in eukaryotic cells is completely intertwined with proteins and histones and all these things, then it takes time to sort of, unpack all this and allow DNA to synthesize. Yeah. So it's maybe not, per se, it may not be the speed of the polymerase, it may just be the speed of DNA synthesis, okay, just making sure that all the DNA is free from the histone before it can be synthesized. And one maybe nice activity which I thought I would maybe show more details on is this little link here, because this is easy to understand that you get the energy of from the pyrophosphate of a new nucleotide to add it onto the elongating chain. But in the nick here, you don't, right? Because what you have in the nick, so you have the previous Okazaki fragment here, you have the new Okazaki fragment here, you only have your 5' phosphate here, and you only have your 3' OH. So how are you going to put the two together? Well, of course you need ATP, right? So you need ATP, and the pyrophosphate is free. And you can see you make an intermediate in which this 5' phosphate is bound to this adenosine monophosphate. And then this phosphate is activated by the AMP here, and this allows the link to be made. And molecularly, this is really what happens. So this is the phosphate here, it's in the other direction, right? This is the 5' phosphate here. This is not so good because they show 3', 5', so it's a little bit strange. It should be 5', 3', but then everything should be changed. So this is a little bit more easy to understand. So this is the 3' end, this is the 5' phosphate that gets activated by the DNA ligase using ATP, so that this 5' phosphate gets bound to adenosine monophosphate here. This is nicely activated, and then you have the nucleophilic, whatever the name, you have the reaction between the OH and the phosphate here, which allows the bond between the previous Okazaki fragment and the new Okazaki. All right, proteins at replication fork cooperate to form a replication machine, yes, because there are many proteins. I've told you there are many, many proteins involved in DNA synthesis. What you get is this. So here you have the leading strand template, and you have the DNA polymerase here that continuously elongate the 3' end. But DNA polymerase is great and can make a lot of DNA. But the problem is that the affinity for DNA is not so big. And if you only have the polymerase, it will make maybe 20, 30 nucleotides at a stretch, but then it will go out. It will leave DNA because it will not stay on DNA. So you must help the polymerase to stay on the DNA until it's finished. And there is another protein which sort of promotes the interaction of the DNA polymerase with the elongated strand, and this is called the sliding clamp. Okay? And it's a clamp that needs energy whenever it's bound. Okay? So what you have, it comes in, and it sort of clamps using energy the DNA polymerase onto the DNA. And then the same polymerase will continue all during the whole synthesis. Then you have another protein. It's called the DNA helicase. Yes, because you also need energy to, to sort of unwind the DNA helix. So this is a DNA helicase. We'll make sure that the double helix is pulled apart. Then, of course, you have the primase. It's a little bit funny, DNA primase. It's making RNA, but so you have this primase here. Then you have another type of protein. They have no activity by themselves, but you can see this is single-strand DNA binding protein. Because of all these Okazaki fragment, RNA primer business, you have single-stranded DNA that is floating around in your cell. So you must protect that. Okay? So these proteins, these single-strand DNA binding protein, they will bind on this single-stranded DNA and protect it. Okay? So you have the primase, you have the helicase, and of course on the lagging strand you also have the sliding clamp and the DNA polymerase that synthesize the Okazaki fragment, starting from the 3' end of the RNA primer. But actually, this really doesn't go like this. It's kind of folded onto itself. This is really what's happening. So you have the leading strand here, you have the DNA helicase here, you have the primase, and then what you get is that this helicase, out of the helicase, there will be the lagging strand will go out, and as it goes out, it will be protected by the single-strand DNA binding protein. And then you have RNA primer here, which has been added previously. Okay, this is the new one here. This RNA primer has been added previously, but then you have the DNA polymerase who has added the DNA after the RNA primer here. So this is the configuration of the, replication fork. But when you see it like this, it's very difficult to understand how this body works. So I think we need a film for that. We need the film for that. So this is, showing it bigger will not really help much. So the DNA will go like this. This DNA will go out of the helicase and will be synthesized. So the beauty of this is everything is close together. Okay? And then when this new Okazaki fragment has been synthesized, the same polymerase can just flip and continue the synthesis of the new Okazaki fragment. So you don't need always to have a new DNA polymerase. Everything is nicely localized, and maybe this. No, yeah, this is just the helicase business. So this is why you need a helicase, because the helicase will really pull apart the two strands of DNA. This is, what could it be?
Clamp.
No, that's the helicase. That's the DNA helicase. And you can see that in the center here, you have really a hole in which the DNA is. And this really kind of twists the DNA around to make sure that it's good. You need a lot of energy because you need to break every single hydrogen bond. So now this will show you how it's mildly. So you can see it's a hexameric protein, and there is a shift of ADP, ATP, phosphate interaction to make sure all this protein sort of, this is mildly changed. The main thing here, I think, so this is the DNA polymerase, and this is the sliding gland. Okay. So what I really wanted to show you is this one here. Because you recognize, well, you can see the film. Green Pac-Man, of course, is DNA polymerase. And with this film, you really can understand why the fork can really progress, in this case, to the right. So this polymerase has just finished synthesizing the Okazaki fragment. Then the RNA primer is synthesized, finished, and then single-stranded DNA binding protein come. Then association of the strand with the polymerase, synthesis of the Okazaki fragment, while this is still being fed through the helicase. And this, of course, continues that way without interruption. And you have many rounds of the same thing. This polymerase stays there, it's always the same, but always the sliding gland needs to come every time to make sure that the DNA polymerase remains. And many rounds of this. So it's easy to understand why the name of lagging strand, because it takes a long time, comparatively to this one. So this is really what's called the molecular machine, right? It's made of many different proteins, all bound together because of random, well, not they have evolved, but because of non-covalent interactions, and that's pretty. And the real structure is more like this one. So we recognize here the helicase, sliding gland, the green thing. And you can see this big complex all bound together. All right. And you got say a great little field. All right. Okay. But you need another stuff, because I'm going to make, maybe I don't need to, maybe it's just a gimmick, but imagine you have, I'm making DNA here. I am making DNA. Can you hold that to me? Keep it like this, okay? Keep it like this. Keep it, don't turn, keep it like this. We are making DNA. Okay, you stay, please. You are Mr. What was the name?
Helicase?
Mr. Helicase, but not only Mr. Helicase, you are also, this is painful. You are Mr. Helicase and you are initiator complex. You are the initiator complex and you are Mr. Helicase. And you are going to pull apart this, because you are the initiator complex, and you are going to make sure that the two replication fork progress towards him and towards me. Okay, so pull this one apart.
That's going to be difficult, right?
Yeah. Only pull. Do you want to try? You can try. It gets more and more difficult. It's easy to open it, but it's difficult because we are maintaining. Okay. And now you can relax. And then pull as hard as you can. So you see, it can, but if I relax, it starts to super curl. You see, this happens. So if we don't maintain strongly, what happens is that these kind of things, and this is bad. You don't want this to happen. So there must be a protein that removes because the chromosome is very long. Essentially, this is what's happening. Okay. So there must be a protein that relieves the tension here to allow the replication fork to progress. Okay. And thank you very much. Thank you. And to remove the tension, this is exactly what's happening. Okay, well, now you have a vivid image of what happens, is that if this continues like this will sort of coil and supercoil, and this will create this kind of bad structure, which is bad, okay? Wow. And to remove the tension that is created by these supercoils, then there is another enzyme that is called DNA topoisomerase. And what it will do, it's a simple nick. One phosphodiester bond will be broken. One bond here will be broken, which will relieve completely the tension of the DNA strand. It will not remove any nucleotide. It will just break one phosphate bond. Okay? And this will make the tension release. And so this one will continue like this. So many times the DNA topoisomerase has to keep making sure that the DNA strand doesn't accumulate too much tension. So you can see you have many different proteins that all interact for DNA synthesis. Okay. And DNA replication, there is another thing that I want to mention today is if you remember the previous lecture, I told you about the different structure in the chromosome, right? You have the centromere, you have the origin of replication, but you have the end of the chromosome as well, which are called the telomere. And the telomere is a very big problem. Because of this characteristics of DNA polymerase that it cannot synthesize a new 3' end. It needs to use one that is already there. So you have a big problem at the end of the chromosome. Because in the leading strand, now the leading strand is underneath there. The leading strand is alright, because you are going to continue the 3' end until you reach the end of the chromosome. That's easy. But the problem is, in the lagging strand, you have your RNA primers there, you have the Okazaki fragments, these will be removed eventually. But at the end of the chromosome, you will have RNA primer there, like this one here. But, I mean, what do you do? What do you do? This is the 5' end. This is the 5' end. So you are left with a segment of DNA that cannot be synthesized because you have no available 3' end. So that's a big problem. There is a gap. And if this happens every time your cell divides, the gap will be broken down and your chromosome will become shorter and shorter and shorter. You can imagine why it's very bad. It's one of the currently the main theory of aging, is that something happens at the end of our chromosome. They always get shorter and shorter and shorter. It's not a completely 100% proof theory, but it's one of the main ones. So to make sure that this does not happen, especially in cells that have to divide many times, like for example immune cells, lymphocytes and things like this, then you imagine this is the end of your chromosome, and at the end of the chromosome, as you know, we have telomere. And telomere, this is very nice because they have a specific sequence. It's always the same thing, and it's a repeat of the same sequence. So you can see this is represented by boxes. So this sequence is the repeat of this sequence, which is the repeat of this sequence. And then, of course, this is the template of the lagging strand. But you have an incomplete, newly synthesized lagging strand here. So you have an enzyme that is called the telomerase. And what this enzyme will do, so this is great, it's a DNA polymerase. It makes DNA, but it contains in its active site an RNA primer. And this RNA primer is complementary to the sequence of the telomere. And so what it does, it will actually use its template RNA to synthesize a longer 3' end. Always it's synthesizing the 3' end as well, right? And it will just make sure that this telomere repeat sequence is elongated so that you can get a full sequence on the complementary strain. And this telomere makes sure that the telomere are long enough to be able to always get the end of your chromosome. So this is what's happening. Some cells have telomeres, some cells do not have telomere. It's very, very still complex to know exactly what happens. As I said, there are some mutations in telomeres that have been associated with aging, but it's not 100% demonstrated. But it's a very important mechanism. If there are no telomeres at all, then your telomeres will always get progressively smaller. And of course, if you have very long telomere, that's okay, because you can get a little bit shorter chromosomes. It won't affect much, because the telomeres do not really have genes, just sequence. But if you lose too much of your telomere, then you will start to lose sequences that are important for proteins or whatever. So telomeres are very important. So remember that there is a specific protein that makes sure that the telomeres are kept long enough to be able to always complete your DNA synthesis, your DNA replication, without progressively losing the end of your chromosomes. And DNA repair will be for next week. I think I'll let you go now.
So that could be studies for achieving immortality.
Well, yeah.
Very long, affect with the length.
Yeah, actually, there are research being done like this. People who are trying to understand exactly how telomeres work and why, some cells with very fast proliferation rate express a lot of telomerase. But it's not the case for every cell. So there are other mechanisms there. But definitely people are kind of looking at a way to kind of make sure that aging can happen without disease and stuff like that. So this is one of the things. Maybe if you can sort of promote telomerase expression and stuff. So this is one of the main theory of aging. So this is the telomere-associated aging, and the other one is reactive oxygen species. So metabolism causes a lot of free radicals, and these free radicals, they are kind of, for example, superoxide. It's kind of special type of oxygen and stuff like that can then react with components of your cells and progressively accumulate damage like mutations and stuff. So it's more like a catastrophic aging thing. It's one of the two main theories. In real, it's probably a mix of both. Okay, so if you have any questions, you can go. No, if you have any questions, you can stay, or otherwise you can go. Why bacteria have no problem? I told you, we have a problem because the end of our chromosome will get shorter. What way has bacteria invented to resolve the problem? Bacteria have no telomeres. This is a eukaryotic invention. So why bacteria have no telomeres? What sort of other mechanisms have they evolved to resolve the problem of end of chromosomes? Their DNA is just like a ring. DNA has no end. It's a perfect circle. Closed circle, so there is no end. So it can continue this synthesis. Anyway, that's the final bit. Thank you very much. Mitochondria also have a completely circular genome, just like that.
They are more progressive than us. If we would have that, that would be great.
That would be great. That would be great. Imagine a huge circle with all our chromosome end to end. That would work. Who knows? Maybe if we are still alive in maybe 2 billion years, this is what might happen.
Yeah, we evolved to
Yes.
or better.
I think it may not work because then, well, it might work, but you need to invent many other things because it will be such a huge circle that it may get broken and everything. So
Yeah. Well, it's possible. It's possible. Thank you for coming on Saturday.
Thank you. See you next week.
Bye bye.
What's up? What's up?
Can RNA primer not attach to very end of 3' end?
Thank you. Thank you very much. Well, it can. It could. But that's the problem, isn't it? Because so if you have the template strand here, right? And then, so this is no problem. You synthesize that guy until the end. But the other way, I want to synthesize this one here. Right? And I have RNA primer here that was just synthesized at the end of my chromosome. And then I made my Okazaki fragment here. But then the RNA primer needs to be removed. And then how are you going to make the Okazaki fragment that completes the end? This is also important, right? And the last RNA primer is here. So what do you do?
Does RNA primer removing and synthesize not occur at the same time?
So what do you think? I'm trying to understand what you said. So,
do different enzymes do RNA primer removing and
no, it's different. It's different enzyme. The enzyme that makes the RNA primer and the enzymes that remove, they are different.
so it seems that the enzyme can remove and synthesize this
So, in the same time, remove and synthesize. So yeah, that's the 3' end. But there are no, yeah, if you could have, if you had RNA, if you had RNA's DNA polymerase, that might work, but it doesn't exist. So you have no protein that can actually, in the same time, degrade. You have no protein that can at the same time degrade and synthesize. This doesn't exist. Because it needs a 3' end and it doesn't have. It's the 5' end here, right? Even of your Okazaki fragment. In your Okazaki fragment is the 5' end right there. So even if you start removing this one, you still have 5' end. So you cannot synthesize anything that way. So well, you can synthesize, yeah, it's the end, so there is no 3' end to continue. So the only way really is to make sure that this is a little bit longer, so that you can have at least one nucleotide here. You just need one or two nucleotides. And then you have a 3' end there that you can elongate. That's the only way. So, Yeah, telomeres are very interesting. Telomeres are pretty interesting. Still many things we don't know. Unfortunately, no. There is no.
I heard that telomere is very high structure, forming a ring.
Yeah, it's just not a noodle. Yeah, it's really everything about DNA is like it's not only just like a string. It has structure of DNA is also very important. Now it's and you have other proteins. You have other proteins that are involved. Not just telomerase, but they are the things. There are a few good reviews about telomeres and aging, and also immune system is also important. Yeah. It's,